All right. Hello, everybody. Good morning and good afternoon. Welcome to today's webinar titled The Automation Conversation, What Pharma and Flow Cores Can Learn from Each Other. I am Sydney Panilla of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Curiox Biosystems. To learn more, visit curiox.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you happen to have any trouble seeing or hearing the presentation. Okay, I would now like to welcome our speakers for today's webinar, Ryan Duggan, Principal Research Scientist at AbbVie, and David Leclerc, Technical Director, Cytometry and Antibody Technology Facility, University of Chicago, and President Glivka. David, I will go ahead and hand it over to you first. You may now begin your presentation.
Well, thank you very much. And thank you for LabRoots for organizing this webinar, and for the good people at Curiox for getting me and Ryan involved. So Ryan, we're here together after the Glivka meeting, which was held in Chicago in September. Glivka is the Great Lake International Flow Cytometry and Imaging and Flow Cytometry Association. And this year, since we were in Chicago, we decided to take advantage of the huge amount of biotechs and startup groups in biotechnologies that are basically popping up all over the place. And the one thing that I thought would be valuable for the audience at the Glivka group, basically trying to figure out what kind of information we be able to share and would be useful to learn from one another. Huge part of that was the automation, and so that's when I approached you a while back to ask you to give a presentation at the Glivka meeting. I kind of wanted to know how you handle automation in your facility at AbbVie. And so I think what we should do is get you started and basically have you give a shortened version of the presentation you gave at Glivka, and then we'll start a discussion afterwards. Sounds good?
Very good. Thank you. Thanks, David. And hello, everybody. And this morning, I'm going to talk about some of the automation workflows that we've implemented as part of our flow cytometry workflow and analysis. And I've condensed this down to essentially the sample handling and staining processes. So what I will not be talking about really is kind of the acquisition and analysis portion today, but equally important things. And I feel like now is really a great time to start to get into automation and at least having those conversations about how to implement them into your labs. And by way of example, I have a few figures here that I put together. The top right chart is showing what a lot of us know who have been in flow for a while, that the number of parameters, the amount of things that we can analyze on a sample has been growing rapidly and exponentially. In fact, this idea has been now dubbed Roederer's law, the idea that flow cytometry is growing in complexity and quantity over time, much like other laws we see in, say, computing and whatnot. And so with the advent of things like the Brilliance line of reagents, now the real dyes, the spark dyes, the Starbright dyes, etc., as well as spectral flow cytometry in general, there is a lot more complexity involved, which could introduce the potential for a lot more mistakes as well. Cytometry cores and the lab personnel rely pretty heavily on manual tools for the most part for taking on these experiments, whether it's staining or centrifugation, preparing reagents, preparing cocktails. A lot of that is still done with manual tools, i.e. pipettes, even multi-channel pipettes. However, other labs have adopted some automation to a larger degree. I'm thinking in the case of, say, clinical labs, or maybe those in industry who have benefited from automation at the tube level. This is usually working with blood tubes and products like the CellMec or the Fax Duet, which will handle blood tubes and do a lot of the staining for that process. But they're usually lice no wash type protocols where we don't have to worry about centrifugation and things like that, as well as other other research cores outside of the flow core, right? So you might see your immunohistochemistry group has an auto stainer. Your genomics lab has automated library prep. Your proteomics lab has lots of robots and whatnot. So the question is, why the automation gap and why specifically flow cytometry? And I'll talk a little bit more about some of the reasons why this might be happening, but off the top of my head, people basically will come up with a few different excuses. One, capital. You need money, you need time to be able to bring these instruments in, which is definitely a real thing, but definitely surmountable. Secondly, it's a little bit too rigid. You might say, in a research lab, my applications are changing all the time, so I can't really standardize things to the point where I'm going to be using the same routine over and over again. And the third one, you might be thinking about job security. If we replace all the people in our lab with robots, I'm going to lose my job, or it's going to impact me in some way. The justification, though, I think outweighs a lot of these concerns. The justification for automation really is about reproducibility, even more than efficiency, I'd say. I'm showing here a table from a publication that is just outlining in a number of different fields areas where we've found concerns about reproducibility. So you can see in different technologies, from genomics to oncology to microarray data and whatnot, the lack of reproducibility not only causes maybe a misunderstanding of the science that's actually going on, but also has repercussions in terms of wasted dollars. There's a quote here that I'll show that I have on screen here that states the lack of reproducibility in preclinical research has an estimated cost of $28 billion per year. And that's just in the US alone, right? If you go into other markets, that would just be compounded even more. So I feel like this idea of reproducible data is really at the crux and the thrust for jumping into automation. Here is just outlining a few of the benefits and perceived limitations of automation. So I'll just run through them pretty quickly. The benefits, again, as I mentioned, reproducibility and reduction in error are always going to be at the top of the list. Just think about the simple task of making an antibody cocktail. When you're just using, let's say, 6 to 8 different antibodies, keeping track of them is no problem. But when you have 40 antibodies that you're putting together into a cocktail, this has happened to me many times, I'm afraid to say, where you wondered, did I just add CD3 or did I add the CD4 yet? Or which one was I actually on? So just keeping track of that, people coming in the lab, distracting you, asking you a question about some other instrument problem or whatnot, may actually lead to a situation where you lose track of where you're at and you either add too much antibody or you miss an antibody. The other thing I think is important is safety. There's really a lot of potential biohazard issues when you're handling these samples. You're walking from your bench to the sink to the centrifuge, sometimes with an uncovered plate of whole blood or something like that. We might be flicking our wash buffer into a sink, which is causing aerosols, all sorts of potential safety issues. that would all be solved with automation. And then finally, for a benefit in terms of a core facility, specifically a recharge core facility, this could open up a new revenue stream as opposed to just relying on people to come and use your instruments or potentially asking you to stain samples without automation. Now, if you can charge for staining samples with automation, that not only increases your revenue stream, but frees up your time to do other things. And of course, there's limitations that go along with this as well. One is, as soon as you buy an instrument, you're worried about obsolescence in the technology. So, it might behoove you to kind of look at technologies that have potential for growth. Sometimes people will grossly overstate the capabilities and benefits of something like automation. For me, I try and temper my either praise or criticism of these technologies just because I don't have enough data yet. And I don't want to oversell the technologies. Yes, there's great things about automation, but there's also bad things about automation too, or issues that pop up that you need to take care of. And then really this concern about worker displacement. I've noticed in my lab, the inclusion of automation has only increased the amount of work for us. As soon as we're able to do these types of things, we get more requests for people wanting to do things with our lab. And that expands to, additional technologies, additional applications, some that use automation, some that don't. So I just feel like this concern about worker displacement may be a little bit overblown. What I'm going to focus on today is really this, like I mentioned, the workflow of the flow cytometry assay. So getting your reagents together, making the antibody cocktail, staining your samples, washing them, and then bringing things towards acquisition. But really, I want to focus on these first two steps here. Once you have your single cell suspension, making the antibody cocktail, and then adding reagents, incubations, and washing steps. So I took the time to actually jot down, as I was staining samples, how much time it was taking me for various actions, as well as how many pipette actions I actually had to do in order to accomplish staining of some samples on a plate. And I was using a multi-channel pipette, so it's not counting every individual sample, but most likely counting every row of samples that I was looking at. And to my surprise, I had over 200 individual pipette actions that occurred over a total of 4.5 hours. 2 of those hours, I had to be present in the lab, right? So even though I was spinning for 5 minutes, I can't take those 5 minutes and walk to my office and check emails. I have to kind of be present anyway. So I took that as hands-on time. Whereas the other 2.5 hours was incubation time. So fixation, antibody staining, things like that, where I could actually walk away and do something else. But this is the amount of time that could potentially be saved. This 2.5 hours or this additional 2 hours that could be saved through automation. And what I wanted to lay out is maybe just my ideal situation of what a fully automated system might look like. And this does not exist yet today, but potentially in the future it would. So let me walk you through it. I'm going to start with the premise that you've already prepared your single cell suspension. You've taken your tissue, you've dissociated it, you have it in a single cell suspension, and you have it in some kind of microtiter plate, 96 well, 384, something like that. So that's done offline from the system. And then what I've dubbed this thing in the middle the automator. So I would load that plate of samples on the automator. And then I would have the automator generate a count plate. So I like to count how many cells I have in my samples so then I can distribute a known quantity of cells to a staining plate. And for that, I use the AO-DAPI method for counting. So I'm asking the automator to generate a count plate that is take a fraction of the sample, let's say 10 microliters of sample, add its 90 microliters of AODAPI, and then I could load that onto a cytometer for counting. The count plate is run on the cytometer, and that gives me a cell density for each of my samples. That then spits that information back to the automator. The automator can then dispense, let's say, 1 million cells to a stain plate per sample. Could be different quantities of microliters per sample to get that 1 million cells. And then the automator is also loaded with a rack of antibodies, with the panel of antibodies that I'm using, as well as titers for each of the antibodies. It uses this information then to make the antibody cocktail to stain the number of samples that I need to stain. And then the automator will do the staining, the washes, the fixed perm, intracellular stain, additional washes, and then finally get the sample ready for acquisition. The plate is then transferred to the acquisition cytometer, and then data could be managed in a automated fashion as well, sending things to an AWS bucket where analysis software or analysis pipelines are enacted from there. So what I wanted to show here is that up until recently, we've been using a bunch of different technologies to kind of accomplish some of these things. So I've listed here on our Bravo, we're using the ability of the Bravo to do some steps like make that count plate and do some of the staining additions and whatnot. We also had a TCAN that was doing our automation of cocktailing of antibodies, and then a product like Lumedix Link that will do the data transfer. But with all this, it's a bunch of disparate instruments that I have to, again, carry plates from one instrument to another. How can we get this all put together in one thing? And really, what is that missing link? And for us, really, the missing link was washing. Washing was kind of the critical step. In every instance that we had, we were taking the plate and putting it on a centrifuge and then carrying it to the sink and flicking it, and then starting our next process, which may be automated at that point. And so in looking at different washing techniques, I had a few that we were looking into. One is just pretty much a standard centrifuge, but has some automation built into it. So this is just a kind of a loading arm that will load samples or plates from a stainer or a pipetting machine into a centrifuge. So that would be pretty similar to what we had been doing. Other methods like this instrument over here kind of automates the flicking process, but requires specialized plates to do that. And then we are introduced to the centrifuge-free washing technology that Curiox had implemented in the Pluto. And not only would this answer the problem or the missing link for washing, but it would also integrate a lot of the other processes like adding antibody cocktails to my plate for washing, for staining, doing the fixation, making the antibody cocktails, the whole bit. And so we saw at some meetings a poster that Curiox had presented where they did a head-to-head comparison on some lysol blood of the Pluto compared to conventional staining methods. And to kind of just summarize what you're seeing here, there seemed to be equal, if not better, staining resolution, and there seemed to be equal, if not better, recovery of cell populations, which was encouraging. And it was enough for us to maybe think about doing a pilot with this system. They said here that it took the Pluto 172 minutes to do the whole process, while the manual methods, the conventional methods, took 138 minutes. So again, just to reiterate here, the automation may not necessarily be a time savings advantage, but it's a hands-off advantage. It's 100% of this time is given back to you to do whatever you need, help other customers and whatnot. So I think that's kind of another take home. The other thing is the system can run 24/7. I cannot run 24/7. So that's another advantage of automation. So we set up to do a pilot study. We had acquired the Pluto HT platform, which the HT just, it means high throughput. It's essentially the full 96 well head compared to the other platforms. And what we ran on there was some mouse splenocytes. And I had 3 conditions. One was just a fully manual condition where I just stayed it how I normally would, centrifugation, flicking, adding all the buffers myself. And then I had 2 versions of a protocol that we had set up on the Pluto HT. And the main difference between them is the condition 3 is the actual increase the amount of washing that we were doing. So in all conditions, the samples were stained with the general immunophenotyping panel, fixed, permed, using the FOXP3 transcription factor buffer set, intracellular stain, and then washed and run on the Aurora spectral analyzer. And what I first want to point out here is, again, we're just like the poster that was presented, we're seeing equal, if not better, resolution of populations on the Pluto as we see compared to our manual methods. And in some cases, you'll notice even for this version 1.3, so the condition 3 where we have additional washing, that in some cases the background seems to be pretty low and the staining intensity a little bit higher. So even potentially a slight bump, but I'm not really interested in splitting hairs on that too much. I just wanted to make sure that it was as good as my manual method, which I can easily say that, yeah, we're getting efficient washing in a completely hands-free operation. And then maybe to our surprise, the one thing that was significantly different is that we're actually recovering more cells after the Pluto washing compared to the manual centrifugation plus flicking of the plates. In some cases, maybe about doubles the number of cells that we're actually recovered. This is, again, this is a single study. I do have technical replicates in here, but it was one study, so I'm not going to put too much emphasis on this. Again, if it were the same in terms of retention of cells and staining efficiency or resolution, I would already be satisfied completely. But to see that we can potentially increase our yield and get better staining resolution makes this even a little bit more attractive for us. Okay, so just to kind of summarize things so far, really, again, the complexity and throughput, I feel, is at a point these days where automation is getting to be a necessity to actually keep up with the amount of demand. And I think this happens both in an academic situation as well as an industry. And so I feel like there's the industry, the flow cytometry industry is responding to this need, which is why we're seeing some more automation offerings being put out by various companies. So we should definitely take advantage of that. And then this idea that it could serve as a new revenue stream for core facilities is something that maybe David and I will explore during our conversations. And I will mention that, this was, even though this was kind of a one-off test here, we've since run a number of other tissue types, including blood and bone marrow and dissociated tumors. And we're seeing equally positive results, right? So we're still fine-tuning the programming of the actual protocols that are run on the Pluto. But I think at the very least, we can say that the washing is working well. The recovery of cell types, even from complex tissues, seems to work well as well. So with that, I will end my presentation portion of this, and then maybe me and David, we can get started on some of the questions. And again, if you have questions, feel free to submit them in the Q&A function. And yeah, that's all I have for now. Thanks.
Can I take my prerogative as a guest to ask the first question to Ryan? And I just wanted to add like a one more point to you. You were asking why is there a gap in or a lag in core facilities in academia in flow cytometry to adopt these um, uh, automated solutions, and we haven't discussed the workload. Like typically, the users of core facilities are students, postdocs that will handle a limited number of samples. Um, Essentially, due to the fact that we're looking at really rare population to begin with. So you're going to pool multiple mouse together and go through all these cells to identify just a few thousands of cells that you're going to pool together to get proper amount of DNA to send to the next. And I so typically my users, whenever I bring up the automated solutions, they feel it's a bit cumbersome. Like one of my users mentioned that just uncapping the tubes of the antibodies and putting them in machines is as much work as actually just doing the pipetting themselves. And so my question, I guess, is whenever we look at these solutions and try to see how they're going to work in core facilities, we might be looking at the wrong, or we might be presenting them in the wrong light. Whenever I presented the Luminex, like the Curiox system before in my facility, we were looking at the throughput and we were looking at, just like how easy it is to use. But then the test when we do a demo is basically you get a postdoc to do a sample on his own, and then we do the same sample in the instrument, and then we compare the data. And the problem there is that typically you're going to get very similar results, right, on the one sample. It's a bit like what you presented. You get better results with the Curiox system, with the laminar flow system, but maybe not enough to justify the investment in time and technology. And so it's the benefit where here I feel is more like it's intangible in most cases, but like I can't help but think of the Superman 3 with Christopher Reeves. And I'm older than you are, so I don't know if you remember anything about that movie. But it's basically the one where Pryor was basically stealing the half pennies from all the transaction happening in the world, and then Superman got in there, and this is the times in different age where Superman was dealing with the white collar crime instead of destroying cities. So it's much better back then. But it's basically that's the general idea. Like you basically have. It's basically using this technology over time is where you benefit. Like it's we're presenting the idea like on a single sample you get better results, which is not particularly true. It's basically the idea that your sample, all the samples you're going to run using this technology will all look better, and so over time the quality of your research in general will be better.
Yeah, no, I get that. I think you've also just proposed a use case of why this would work in a core facility that handles lots of samples compared to a single lab that maybe only does 10% of those samples. Meaning, right. if I'm in a core facility and I have a service whereby I receive samples from, let's say, clinicians who have an interesting cohort of patients to look at, and they don't have the lab space and they don't have the technical expertise to do this, but so they just want to drop off samples and partner with me. Now I could take all those use cases and use automation to handle all of that influx and do it efficiently and reproducibly. Whereas if I were a core facility that was, let's say, not doing staining, and I just had to set up as an instrument, standalone instrument that people come in, perhaps the value would not be appreciated as much in that case, right? Because it would just be one person at a time coming in to use it. They wouldn't really see the use, the aggregate use over time and number of people. But if you're a core facility who is handling samples, or maybe you want to start doing that, yeah, it seems like this would be a great fit for that.
Okay,
Yeah, go ahead. Sorry.
perfect. No, that's okay. I was just going to go ahead and jump into a few more questions that we've had come in, if that sounds okay to you both. The next one that we have here says, what's the temperature control like in the Curiox?
Yeah, so there are a number of stages on the Pluto HT. That's the one I'm familiar with that will keep temperature from 4 degrees to 37. So cold or warm is possible. And that could be also programmed into the routines. So you can turn that on and off as needed.
Okay, perfect. Any comments on SciTech Orion reagent cocktail preparation system?
I'm not familiar with it. I haven't used it. I mean, I'm familiar with it, but I haven't used it. So I can't really speak to that.
Okay, the next question that we have here says flow cytometry continues to evolve, but the expectations on labs, both pharma and academic, have grown faster than resources. How do each of you approach scaling workflows without increasing complexity or cost?
It's a
Any
good
ideas,
question.
David?
Yeah, so I'm a bit puzzled by the idea that the so let's put it this way: the whenever we think about our workflows, the new solution, the new evolution from the field is coming into most likely the technology we're going to adopt are most likely going to ease up the pressure in a way. Like flow stompers, the evolution in the last many years have been spectral. Spectral is awesome. Uh, like sorting in cartridges is great. We new ways of manipulating cells. Those are two big ones. The third one is just the interface with the instrument have gone leaps and bounds. Like it used to be incredibly hard and very perilous to run samples on a flow stamper. Different ways you could mess up your acquisition. And nowadays, all these machines are like you can just put your sample on, and the machine already has a whole set of values or in features preset, and you the data at the outset is great without you having to break your clothes all that much. So every time we get a new technology, it basically improves the service, and so increasing complexity or cost. Like I would say, for example, in the case of automation, we've started applying the automation tools to multiplexing assays, and basically instead of doing like getting two of my staff to run a multiple number of kits, we now just run that on a single instrument, and everything is done like basically it's automated. The results are great, and so we save a bunch of time for my staff, a bunch of money in reagents, loss of samples, stuff like that, and so yeah, I I think that's the evolution of technology is just a plus for us. Like we just need to take advantage of these new tools.
Yeah, and I would just add the fact without increasing complexity or cost. I don't know if that's necessarily realistic because I think increased complexity is a natural evolution of that, but I don't think that's necessarily a bad thing. It's increasing complexity in a smart, efficient way that uses automation to your advantage. So it might be a very complex experiment, but a robot's doing it. That's not going to mess things up. So that makes it easy.
Okay, perfect. The next one that we have here says, what factors make automation feel approachable for scientists and core users? Is it software design, user familiarity, or the ability to follow SOP like logic?
I feel like
Yeah, I think
Ryan used
the
to talk about this. Sorry, go ahead.
yeah. Well, I was just going to say, yeah, the SOP like logic I think is very important. It's at least important to say I can check the boxes and say this process is doing the same process that I would do, and achieving similar results. That's like the very baseline. So much so that we find ourselves watching the robot do its work, which kind of defeats the purpose, right? But I want to just see that it's doing all the things that I asked it to do, and it's doing it in the same way that I would do if it were my hand compared to this robotic arm. Once I do that, and I'm confident that the system will do what it needs to do, that's when I think there needs to actually be a change in mindset and not think as linear as humans do, but to allow the system to add efficiency and add the steps in the right order that maximize efficient processes. And this might even be something like making it AI enabled, such that I can just talk to it like a normal human being using normal words. So I could just say, "Hey, I want to stain with my T-Reg panel, and I got 30 samples on this plate," and it kind of fills in all the gaps and does those things. So I think that will make it more approachable once it's kind of like our own personal Alexa or Siri built into the automation. But yeah, I think initially, it has to do what I tell it to do, and do that without error.
I agree. Like you used to say that about the flow stampers, like the most important. Like at some point, it became kind of clear that most platforms on the field would essentially provide similar results in terms of like looking at lymphocytes, and so the main difference became like how do you put your sample on the machine was the most important thing, and the rest is all. And this is kind of the similar idea. Like once the machine is working efficiently, the system is providing SOP like workflows that are easy to handle, then just the way you direct it to do the work is the main difference.
Okay, great. The next one that we have here says, can you add filter step to the workflow for more sticky samples?
Yeah, certainly. So there's actually two points where we currently do this. One is before the samples are loaded onto the Pluto, they are passed through a 96 well plate filter that has a 45 micron mesh at the bottom of it. So it's just a quick pulse on the centrifuge to pass the samples through that filter. Then that goes on to the Pluto for the automated staining. And then because of steps like fixation, where potentially you can get more clumps or just things settling and clumping, we then have it finally at the end of the staining process, move the cells to a filter plate, and then they are again pulsed through the centrifuge and then loaded on the cytometer.
Okay, great. What fixation buffer have you tested, and which are compatible with Curiox?
Yeah, so most commonly we use the transcription factor buffer set from Thermo Fisher. That's the one that's used commonly for FOXP3 staining, but can be used for a variety of things. We've also used the lyse/fix buffer with success, as well as just general formaldehyde PFA, 2% PFA fixation. I do know, and maybe you'll have to follow up with Curiox at this. I thought I've heard or was aware of the fact that the alcohol-based fixation, like methanol-based fixation, may not be compatible with the washing method due to settling of cells. I could be wrong on that, so don't quote me. But yeah, if methanol fixation was an important step, you probably might need to do that offline.
Have you looked into the dishwasher soap fixation method yet?
I have not tried that. No.
All right. It's something that came out.
Well, yeah, we'll have to put that on the list.
Yes.
Okay, David, this next question is for you specifically. It says in academic cores, budgets and shared usage are constant realities. How do you weigh the trade offs between investing in automation and maintaining accessibility for your users?
Um, so I think when we try to, so we've been looking at automation for a number of years with various degrees of success. When we think about automation, we are essentially looking at uh, improving the workflows that are currently problematic. And so the only one that came up at this time has been that we've been successful at leveraging to get equipment has been the multiplexing. But now that we're looking at more and more complex spectral panels, we think that developing the workflows that will help people whenever they prepare their FMOs or their antibody cocktails and whatnot. I think that's going to be something that will definitely be useful for users. So maintaining accessibility for users and investing in automation. At the end of the day, our users want to publish papers, so we do have to provide enough instrument time. But instrument time is composed of a bunch of experiments that are successful, but a bunch of experiments that fail due to a bunch of reasons, including bad sample preparation and stuff like that. So actually presenting tools that will improve the quality of the samples should actually result in getting more effective time on the instrument. So they kind of go together. It's kind of hard to quantify and justify that in a in a spreadsheet that I'm going to present to my supervising committee. But I think they kind of go together, and you can't just bet on more time on the instrument. At some point, you need to think about how you're going to prepare the samples.
Okay, great. The next one that we have here says, I am curious about the permeabilization and fixation staining steps, then following the wash step. How do you deal with most of your cells not settling after this? Ultimately, do you lose some cell samples?
Yeah, I'd say, practically speaking, in the fixation methods that I described earlier, we're not seeing a um, specific loss of any cell types. So the ratio of cells that exist compared to our manual methods are retained after fixation permeabilization. It doesn't seem like, well, so I would say in terms of settling, and again, this might be a follow-up question for the technical folks at Curiox, but um, since you're using an automated platform, time is kind of a luxury that you can play around with, right? So if you're used to doing a fixation for, like, let's say 30 minutes, because it's just sitting on there and who cares when it finishes, leave it on there for an hour, let it settle for 2 hours. What does it really matter? If it finishes at 2 in the morning, it's fine. It's temperature controlled, it's light controlled, it can sit there, it's fixed. You're not going to run it till the next day anyway. So you can definitely play around with all of those to perhaps ensure you have the highest recovery possible. But yeah, I haven't noticed any issues with retaining cells specifically in fixed protocols.
Okay, great. The next question that we have here says, when considering new technology, what defines affordability for you? Is it cost, efficiency gains, or the ability to free up skilled staff for higher value work?
I would say for me, yeah, I value kind of that freed up time for skilled staff. Um, cost is a funny thing, because it's, this is going to be a capital cost. So budgets for that are different. There's different avenues for acquiring that. That I think is a surmountable um, feat if you needed to try and scrounge up some money somewhere. But you can't really. Um, there's no real replacement for that additional time that somebody might acquire to be able to do something else, to you know um, even do things like professional development, right? If you need to free up time from staining and now you can learn Python or R and gain some bioinformatics skills, I mean, that would be a hugely valuable offset for the time that you saved by not having to sit there pipetting in a lab. It really irks me when I see people sitting at their bench for hours and hours, pipetting up and down. It just seems like we should not be doing this stuff anymore. I get it; it's something that needs to happen, but I would be all for completely removing those steps from any of my technical staff in the lab and having them spend more time on things that can actually matter for us in the end.
I'm gonna go with cost, and mainly the cost of the samples themselves, which typically we don't put a price on because they're coming from all over the place, and they can be very easy to prepare. Sometimes they're incredibly hard to come about. But there's the technologies nowadays, especially in flow, have improved in such a way that you can now run experiments where, like I said before, because of the interface, because of the way the machines are built, you put the samples in the instrument and the data came out properly, and everything is usable for the downstream analysis. And when I look at the technology we're going to adopt in our facility, I'm basically looking for all these new tools that will allow us to process these samples that might be incredibly expensive and hard to get by, and in a way that will minimize the chances of making mistakes, poorly staining the cells, and all these things. And so in that sense, like, the automation tools are just like on top of the list. That it's kind of hard to justify to users who are used to it. It kind of requires some kind of a paradigm shift in the way people think about how they prepare the samples. But I'm pretty sure that a very, very good case can be made that automation is a preferred way to run even a few rare samples in a way that will be more efficient.
Okay, perfect. It looks like this next one specifically is for Ryan. It says, I have a question about your AO DAPI counting method on the cytometer. I've always heard that AO is a very sticky dye. Do you need to use a special cleaning protocol after using this dye on the cytometer?
Yes, sounds like somebody's speaking from experience. Yeah, acridine orange is very sticky. On the on the Pluto, I'll just mention, if and when we use that, it's disposable tips, so that doesn't really impact things there. But yeah, on the cytometer, as you mentioned, it can be pretty sticky. I found that doing the kind of a bleach protocol, running a sample of bleach followed by some water. We run this most commonly on our Quantion or Pention. And in that case, they have a built-in fluidics cleaning protocol that basically runs through bleach and then water, and that takes care of things pretty well. But on other instruments, like on the Aurora, yeah, we just do the flow cell cleaning protocol, again, with bleach and water, and that seems to work out. I remember, I don't know if Dr. Darzenkevich of happy memory, had the idea of running caffeine, so coffee, essentially through your flow cytometer to remove some of these dyes. I'm not sure if and how well that might work with AO specifically, but I've got plenty of coffee around, so we could always try it and see if it works.
Was there a reason you use AO specifically, or could you use some other?
You could. I just find it's so the only requirement is that it's a nuclear dye that stains both intact live cells as well as dead cells.
Okay.
So any dye that fits that bill could be replaced.
Okay. Next one says, when introducing automation to staff or collaborators, what training elements or usability features make the difference between hesitation and buy-in?
I think it goes back to what I was explaining before. We tested the Curiox system many moons ago, and I don't think we presented the right features to my to our PIs. Again, doing a simple test, one sample from a PI from a student, and the other sample by the instrument will present data that are very similar. You really need to think about what are the really important features that you want to see improve. Ryan presented pretty awesome data in terms of the number of cells that are that remain in the samples when you use the Curiox platform. That is like it's a 2-for-1 difference between centrifugations. That's kind of amazing. So again, just look at what the workflow, what the problems people are having, and try to see how that technological solution is solving the issue, and focus on that.
Yeah, and I would add, I try to insulate um, end users or collaborators from new technologies until I've vetted them well enough that I'm pretty satisfied. So I'm not going to like throw somebody on the Pluto the day after we install the instrument, right? I'm going to run through all of my battery of tests, try my hardest to break things on the instrument, and then say, "Oh, okay, I'm at least satisfied that the technology works as advertised. Now I'm going to start bringing people in." And I think my users appreciate that, and they know that about me. So they know that when I've finally decided on an instrument or a technology, that they can be somewhat assured that it will work for their use cases. And it's not just the new shiny thing that we have in the lab that we're just going to throw everybody on. So I think building that relationship of kind of honest evaluation of new technologies and telling people both the bad things and the good things that can be expected when using this instrument is important. It's important to do that.
Okay, great. Next question here says, when introducing a new platform or workflow, what builds confidence amongst users, especially those who may not have prior automation experience?
Yeah, I think that's, kind of some of the stuff that we talked about already. I think maybe even it could benefit people to enter slowly and cautiously into automation. You know, perhaps if you have the luxury of having different types of instruments around, even not necessarily in your lab, but like visiting your genomics core and just seeing how other people have relied on these technologies for years now, that might also be a prudent step in introducing automation to new users. Just to say, hey, this isn't something that was invented for flow cytometry 6 months ago. This is technologies that have been present in clinical workflows for years now. And a lot of lessons have been learned to get us to this point. And you can kind of rest assured that this is an instrument and a technique that is not going to just ruin all your samples as soon as you put them on there.
I'll repeat what Ryan's point about vetting your instruments before you put your users on them is critical here. There's nothing that will sync an instrument faster in a core facility than a couple of people trying to run an experiment and failing, and then they talk to each other, and they just next thing you know, nobody wants to use that platform. So you really want to, whenever you put somebody on, be sure that everything's is set for success.
Okay, great. What was the reaction from your colleagues when they saw it in your lab?
Yeah, I think it's a very impressive instrument in size, certainly, and just kind of the expansive layout of the deck. I mentioned we have other automation instruments in the lab, and so they're significantly smaller. But I think everybody's very excited about the potential that we're envisioning for the Pluto, and eager to get started. Again, knowing that we've invested in this platform gives them some confidence that we think this is going to be useful and work for their application. So yeah, everything has been positive thus far.
Okay, great. The next one that we have here is for you specifically, Ryan. It says in large R&D organizations, scalability and reproducibility are key. How do you evaluate whether an automated solution can maintain reliability across teams or sites?
Yeah, so we do quite a bit of cross-site collaboration, and both in terms of samples being shipped from one place to the other. But yeah, I think automation is one of those things that I think will only help allow that parity to take place across sites. Um, it's instrumentation, like let's say the Aurora for spectral analysis. A lot of work has been gone into those instruments to kind of put them all in the same state, such that running your samples on one instrument here compared to another instrument on the West Coast, you should get comparable results. You can confirm that using various QC, QA techniques. But the one thing that has never really been normalized is the actual sample staining processing part, whereby different people's pipettes or pipette technique or washing technique is not standardized. And so taking that out of the individual user's hands and putting that into some kind of automation surely will go and lead to better reproducibility across sites.
Okay, perfect. We will go ahead and wrap up here with one final question. And this one says, many systems now emphasize SOP aligned interfaces. From your perspective, how important is it that automation mirrors how scientists already think and work at the bench?
Yeah, I'm actually hopeful that they don't work like I work at the bench. Again, just this idea of I think in a very linear fashion. I do step one, then I do step two, then I do step three. I think having machines and having algorithms that optimize things and can actually multitask, because we may say we're multitasking, but we're not. But that can actually multitask, I think opens up a whole nother level of efficiency and possibilities for us.
I agree with that. The just a problem to think about, if you have, like, say, an experiment with 20 FMOs to prepare, there's a really stupid way of preparing 20 FMOs, and there's a really, really smart ways, which is probably you have to think about the way you're going to pipette all these antibodies in the tubes in the most efficient way. This is something that AI will probably resolve in very few minutes, where you might spend a week and months trying to figure out what the way to go. So I agree with Ryan, and let the technology support us.
Okay, wonderful. Well, thank you both again for your time today and for your very important research. We would also like to thank LabRoots and our sponsor, Curiox Biosystems, for underwriting today's educational webinar. Before we close out today, I would like to thank the audience for joining us and for their interesting questions. Those that we did not have time for today and those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. As a reminder, this webinar can be viewed on demand. LabRoots will go ahead and alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. All right, until next time. Thanks so much, everybody. Bye-bye.